And again, Lisa, thank you for organizing this great meeting. This is the third one for me, I believe. And it's great to see the families again, the same faces. And so what I was asked to talk to you about is our ongoing gene therapy trial, and I think there's a lot of excitement regarding this work on multiple levels. So moving forward, I just want to acknowledge the team because I may run out of time. No, just kidding. So as you can see, there's great number of people involved in this work, and it's actually growing as well. So we started this with an Age Grant at UCSF. This is a university setting with big medical center. And people here are listed that have been involved in this. And this is 15 years in the making, pretty much. I mean, I'll give you some flavor for how this was developed over time. But Nalin Gupta is the lead neurosurgeon with Paul Larson, who's doing a lot of our Parkinson's gene therapy trial. Anna and Amy are the neurologists at UCSF. Amy actually has moved to WashU. And then we have Jill Imamura, who is just incredible in terms of running the trial for us. And I'll try to get this, because they don't have a pointer here, it looks like. So there we go. So basically, the person that you will hear in a minute is Toni Pearson. She started this work with me when we were still with Daryl DeVivo. And now she's in Washington University, very much involved in this work. So she will give you actual update on some of the neurological issues as it relates to these patients. Kathleen Black and Erin Smith are on her team in Washington University. Keith Highland, without him, we wouldn't be here. I mean, this is really a seminal work that he has done, and Simon. So it's a very important biomarkers, and you'll see some of the data as well. In Ohio State, we just set up a site there with Raz Lonzer being the clinical PI over there. And then at UCL, I mean, this is I don't know, man, just 7 years. We've been trying to do it. Unfortunately, Manju was not able to secure funding for the patients here in London, but we hope to be able to treat the European and UK patients with Manju's help as well. And then we have advisory board, a great group of people that's been advising us over the years. So I'd like to recognize them as well. So here's something that I think is of interest as we move forward, and this is Ted Friedman, who in 1972 said this, that deliver healthy DNA to treat human disease using viral vectors to deliver the therapy to all cells in the body. So that's what, 40 years ago or so? And that's about right, I think, in terms of establishing a technology that can be really life-saving. So I really have to say that being in this field for close to 25 years, I mean, we've seen a lot of upswings for multiple different reasons. Some of this was not necessarily science-driven, but like any technology is going to take a long time, and it's going to take experience of many people to get it right. And I think it's a process. And I think you've heard me speak here 6 years ago or so, and we were not obviously as experienced as we are right now. So what is gene therapy? In this particular context, I mean, what we're trying to do is to deliver this microphone is awful, deliver the virus that is being completely gutted out, and it has a healthy version of dopa decarboxylase. What's important for you to understand is that these are nanoparticles, so it's not small molecules that we're trying to deliver, and we have to put it directly into the brain because these particular vectors would not efficiently cross into the brain. So we had to develop a surgical technology to do it. And there are a couple of things as it relates to that. One, of course, there's a physical size to it, so you cannot just take a needle, inject it into the brain. It's just not going to go anywhere. The other thing is that these particles have very different receptors, so you have to fully understand how they bind to the cells and what happens to them. Because in some cases you may infect wrong cells, and in some cases the expression may be temporary. So all of this really had to be worked out, and here's just to give you a flavor of these particles we're using. This is a actually physical picture of these viruses. They're around 25 nanometer large. And there are many different serotypes, and they do different things in the brain. And so you have to really understand, how to craft them for a particular disease. So in this particular case, we've been working with the serotype 2, which is highly neurotropic, and also has other properties which I'll describe in a second. Those other serotypes do similar things but differently, and they are not as well characterized, I believe, as this very original AAV that we've been working with for a long time. So vectors, and now how do you deliver these things to the human body? And you have basically three choices, and one would be intravenous. So some of those serotypes, for example, AAV9, could enter the brain. In the best case scenario, you can get maybe 10% transduction, but it's mostly cortical, so it doesn't really go into deep parts of the brain. Then you can put it into CSF space, so you can inject this directly into different compartments. And again, it's a more systemic administration in the sense that you will get a global expression. Or you can put it directly into the brain, and this is what I'll talk to you today about, because this is, in our opinion, the only way perhaps how we can get this to express in very specific parts of the brain as it relates to AADC deficiency. So this is what started this whole journey for us. This is 15 years ago when we treated first patient with Parkinson's disease at UCSF. It's maybe like more than 15 years. And this was of the same gene, trying to develop a system in which we can give patients a much better way to respond to levodopa. The big issue at that time, and still it is for many of us, is how to deliver it. So here we have an example of how we did it, and we call it the version 1.0. Close to 20 years ago, we've developed this based on what was available at that time, which is a stereotactic frame that you attach to a patient head, and here you can see two cannulas going into the putamen. So we really didn't have much understanding what's going to happen next, and here is one of the fellows at that time that sort of looking at this whole thing saying, what the hell is happening here? I mean, seriously, I mean, you put two cannulas into the brain, and what happens next? In other words, where will those particles go? Would they cover enough brain to really make a difference? And it really didn't. This trial, although safe, didn't yield any significant clinical data. So I was a little disappointed. At the same time, this technology was transferred to Japan. They've done, how many patients did you do? Parkinson? Yeah. So, that was pretty much what was adopted in Japan and pretty much used for the AADC deficient children. So they basically adopted that version 1.0. So as we move forward, we've realized that, to do it in a more efficient way, perhaps, would be to do two things. One would be to get safely to the target. This is obviously critically important, and the use of those frames doesn't really let you do any real-time navigation. So I think the issue with targeting it's been a difficult one. And the other one is, of course, can we monitor the gene delivery? Can we see what we are doing? Can we somehow rationalize the gene therapy delivery? So here you have the system we've developed at UCSF that actually allows us to operate on the patient in the MR so we can see in the real time what's happening, both for the trajectory, but also for being able to adjust at any given time those trajectories to give us really very great degree of accuracy. Around 400 some patients been treated this way for different indications with a resolution of perhaps 1.2 millimeters. And here we have an example of other things that had to be created, such as sterile fields, working in the magnetic field with metallic instruments, things like that. So there's a big issue of patient safety that had to be worked out. And here we have Nalin Gupta that is infusing AADC into one of our patients. So again, this was probably 10 years in the making, trying to get to the point in which we can really, in a very high precision, deliver this gene therapy. So you'll see some more of it in a second. One of the software had to be developed for that, so again, allows to adjust the trajectory in the real time. And here we have an example of software telling us how to turn little knobs to really get adjustments within a millimeter or so. So that's the first part. In other words, can we deliver it safely, avoiding any blood vessels, of course, and can we target it where we really intend it to be? The other one is the monitored gene therapy. And the issue here is just what you see here on this landing strip. If you miss it, you're going to get into the ocean. And the same happens with the fluid, because if you don't get the right distribution, in other words, you miss the runway, things will leak into the CSF. And there's great opportunity for that to happen through perivascular space, for example, or also through just infusions that go into the lateral ventricle, for example. So it's important because, you know, if we have planned to cover a certain part of the brain, we have to see what we are doing, of course. So that's what this was meant to do. The third thing, which we worked on number of different trials, it was this concept of convection-enhanced delivery. And you know, it's incredibly simple if you do it right, but it can be devastating if you don't do it right. So here you have an example of, and I'm going to play it for you again, of us infusing a blue dye into agarose gel. And what it is really, it's a mixture of gel with water. So you can see this blue thing growing very nicely, and it pushes water from this gel to the surface. That's how it works in the brain. You're displacing the extracellular fluid with the fluid that now carries particles. If you don't do it right, you get what's called the reflux. And that has to do with type of devices we had to develop, such as this one, with a specific step design to really prevent that from happening. So I'm just giving a flavor of all these little details that had to be developed to get this thing right. So here we have an example now of the infusion, just like in this gel, into a monkey brain. So we've done it very carefully over the years, and now you can see instead of blue dye, which you are looking at, it's a gadolinium that's been infused into monkey thalamus. Similar thing, we're displacing the extracellular fluid and displacing it with the therapeutic agent. So both things you can see. Of course, human head doesn't have, you know, glass jar for you to see this, so we had to obviously use the MR technology, but the endpoint is the same. We know what we're doing, and we can react. During the surgery, we can change those infusion volumes, rates, place the catheter differently, can be very proactive to get it right. The other observation which is relevant to this is that when the convection is done correctly, you achieve, as you can see here in the EM, this is frustrating. Okay, here we go. Here what you may or may not see, it's the brain that has not been treated with the convective delivery. And you can see a huge difference in the extracellular spaces. So we can open them by creating a pressure infusion that creates little gaps. And those gaps are big enough that you have very efficient flow of the nanoparticles, but also gadolinium tracer, leading us to this, which is a complete co-distribution of gadolinium and the gene expression. So here I have one of the examples for the AADC in the brain. Here's an infusion. Here's an expression. And this again in the monkey brain. So we have a tracer, and this is true for multiple genes. This is, for example, for our ongoing trial with GDNF, where you can see the tracer and then the expression. The interesting thing on this slide is this, which I won't have time to go into, but it's the leakage from the putamen that you see both in monkeys but also in patients, that if the catheter is placed too closely to a perivascular space, your old vector is just going to leak out completely. Happen in multiple cases. So here's version 2.0 that took us a while to develop, and this is the first patient that we've treated in 2013 with GDNF, and you can see the putamenal infusion of that particular patient. So it was great to really validate this and get a lot of confidence that the system actually can allow us to see gene therapy. Here's the reconstruction of those infusions, and just for the reference, we infuse 450 microliters into each putamen. So it's a probably abstract number for you, but it's the volume that has been infused. Now what happened next, if you look at what happens in all these patients, the green is the putamen on both sides. So there's our target in Parkinson's disease, not in AADC deficiency, but in Parkinson's. And then you can see that a lot of these expressions are actually outside of the putamen because there's tendency of fluid to backflow. We had to change the design of the catheters to keep it in. But the message here is that we have only partial coverage with state-of-the-art technology, right? So convection, imaging, catheters. This is the best you can get from 450 microliters using this approach, as we call it, transfrontal approach. You can also see these little rivers here flowing to the medial cerebral artery through the perivascular space. So again, a lot of vectors going to leak out. We know that because we can see it. With all that effort, all we can get is 22% coverage of the putamen. That's all. And we put 250 microliters into the putamen. And you cannot get any more with that kind of coverage. So, we had to think hard, talk to the FDA multiple times, and they allow us to do something very unusual for them, which is they told us, well, why don't you adjust your delivery to maximize the distribution? Again, it's a safety study in Parkinsonian patients, but they've realized that it's all about the coverage. That if we claim to cover a specific part of the putamen, we have to be given a freedom to adjust that volume. And here is version 2.1, if you will, on the patient now that gets close to 800 microliters. And you can see 3 infusion sites being approached in this particular subject. And again, we make those decisions during the operation. In other words, we don't see good coverage in one site, we'll reposition the catheter, deliver more. Again, the effort is to cover the target. So here's that particular patient. So the other thing I want to share with you is that this is the shape of the putamen. So it is quite elongated structure. And with going vertically, this transfrontal approach, you don't have much runway there. In other words, the opportunity to cover the putamen using this approach is really poor. We came up with the approach now that allows us to go in the long axis of the putamen. Again, effort being, can we minimize the surgical time and increase the coverage? So that's shown over here. In other words, this is what's called the transfrontal approach. This is what we call the posterior one. And it's pretty phenomenal what you see here, because from one infusion site, now with going to 1.5 ml per infusion, we can get this. In other words, almost complete coverage of the putamen, 60% or so, because we actually were able to adjust the trajectory right. So why am I talking about it? Because I think it's important for a lot of you here and families that have expectations as it relates to gene therapy, that you understand the issue here. Because we didn't understand in the beginning with Parkinson's disease and didn't really get much improvement in those patients. But here, we're actually pretty happy because you can see the different cohorts of Parkinsonian patients. We've got 30-some patients treated. This is the patients that we deliver 450 microliters. You get 21% coverage. We go to 900, we get 34. Then we increase it even further, like 1.8 perhaps, we can get up to close to 60% coverage. Why is it important? Because of this. The clinical improvement in these patients, it's absolutely volume dependent. So if you don't get good coverage, we only get 13% of coverage, an improvement in the patients in terms of the PET scan. In terms of the clinical improvement, it actually didn't do anything. You double the volume and coverage, you get a huge improvement in the clinical response. And again, it's volume dependent as well. So, to me this is very important because, those lessons we learned from Parkinson's trial are incredibly valuable because, we had to work it out in that trial before we felt that offering this to children was responsible. So I think that taught us a great lesson. Here's another lesson, which is, remember this jar I showed you that this blue thing is just expanding? So what you measure right now, you can do it now in the patients. You can find out the relationship between how much we infuse, the distribution, and how much coverage we get, right? So let's say I infuse 100 microliters, and I'll get 300 cubic millimeters of putamen covered. It's 1 to 3 if you do it right. Typically, it's around 1 to 2. So that's what this graph is showing, because this is where I think the experiences with gene therapy in Parkinson's has taught us tremendous lesson in terms of the putamen. So the volume of one human putamen, and child putamen isn't that much bigger, is around 4,500 cubic millimeters. So that's the size of your target. So if you do the math, it's very easy, because if you deliver, let's say, 100 microliters, as we did in the first PD trial, we didn't cover much at all. It was perhaps 10%. Other trials, this one also failed. It was 150 microliters. This is a trophic factor given. We started our AADC and PD trial with this, and I showed you already data from the first group that didn't really improve. PET improved very nicely, but not clinical outcome of those patients. So, and there, we've covered 22%. Then you go to 900, there'll be around 40 to 50%, and then those larger volumes can get us up to 60%. So I have a big problem with the trial done in Taiwan and in Japan, because, again, they're using the version 1.0 that we've introduced 20 years ago, and they're delivering 160 microliters. So by definition, they'll get 15% coverage at the maximum, not anymore. And here's the size of the human putamen. So it's a big issue, because I think, as we've learned with the Parkinson's trial, if you don't have coverage correctly, you're not going to get good clinical responses. And here's the work that was presented, the group in Taiwan in May. And I think that what they're reporting huge variability. 5 out of 15 gain head control, 4 out of 15 gain able to sit unassisted. These 18 patients. I think has to do with sort of hit and miss in terms of the distribution. You know, we would like to see consistent responses, you know, very dramatic clinical improvements. But I think learning from Parkinson's trial, I can tell you, I'm very convinced that not being the case because of the volume issue. So that's one thing I wanted to talk to you. But the other one is, the other big aspect of the gene therapy that has to do with how brain is wired and how these particles do work. And so what you're looking here, it's basically a situation where we do believe applies in human brain, of course, and spinal cord, which is a lot of projections. So we know about all of this. I'm going to show you some of those that are relevant to the AADC deficiency. So this is called Dti Diffusion ultrasound imaging. And here, you can examine the pathways. And for example, in Parkinson's disease, there are certain pathways which are degenerated. So the question is, are they also not present in children with AADC? So we'll talk about it in a minute. But I'm mostly using this slide to illustrate that not only delivery matters, but also what happens to the particles when you put them into the brain. Clearly, something's going to happen because they bound to the cell, they're being incorporated into the cell body, and cells have axons and dendrites, things move, right? So you have this illustrated here, and you can see those pathways quite well. So the main thing, you did see this slide today already, so I guess we got it from the same website. It's the fact that, you know, with studying those different serotypes, we realize not only the serotype, but the production method and purification has tremendous repercussions when it comes to how things move along those axons. So in this particular vector, when we put it into a cell body, let's say in Parkinson's, it's going to be a medium spiny neuron, this vector is going to move anterogradely, so it's going to go down. So in Parkinson's, it's going to go to globus pallidus, subthalamic nucleus, reticulata, for example. So we described that pretty carefully. But this is what we call the Anterograde Transport. So if you put in the cell body, and because of the surgical targeting, in other words, you know, we target specific parts of the brain because we can use MR guiding and can place the needle and be able to restrict that primary transduction to a specific part of the brain. So it's shown here. Again, you saw this slide before. We also infuse at the same time AAV2, similar to what we're using in children, that carries GFP. This is a gene, it's a marker gene called the green fluorescent protein. So here you have an example of co-distribution between the tracer and there's only one site that was infused against the monkey brain. And the next thing you see this, which is tremendous axonal transport into the cortex. So it's called Anterograde Transport. So what it means is that if we target correctly with this vector parts of the brain, we can depend on this Anterograde Transport. So we've described it as 10 years ago, I think. And then decided to do the same thing in this particular clinical trial. This is the wiring of the brain that is relevant to what I'm going to just about to tell you. So here is this thalamus and the projection to cortex called thalamocortical projection. So you can appreciate how incredibly efficient that system is. Now we have the other projection, which we had to really figure out if that projection actually exists in AADC deficient children, that we could possibly use to distribute the gene therapy from an area of the brain that actually projects to what we call the striatum or neostriatum, which is putamen caudate So that's with this particular projection called nigrostriatal pathway. And then we also decided that we'd like to target it's called the mesolimbic pathway, which is not only responsible for some of the motor improvements, but drives cognition. It drives a lot of things which are not motor related. And I think you will see from Dr. Pearson some of the results from targeting that pathway. But again, in designing this trial, we realized that if we target this cell bodies in this part of the brain called the midbrain, we could use the axonal transport to really spread the AADC very beautifully throughout the whole target area, which is in this case putamen, caudate, nucleus accumbens, septum, cingulate gyrus, and prefrontal cortex. So these are critical areas in the brain that don't have any dopamine in children. So again, you cannot really reach any of this if you do multiple injections into those structures because it's physically impossible. But natural axonal transport will deliver it there. So that was pretty much the first prospective gene therapy trial that is using an axonal transport to deliver the gene product. I think the other thing I want to make sure that I tell you, because I think it's very important, is how this is supposed to work. So here we have a synapse, and you already heard today about two major components of the system. One is this projection from midbrain region, called the nigrostriatal system, that normally in our brains is carrying little vesicles with dopamine, because it is made in the dopaminergic cells, packaged, transported into the terminal region, and then in a highly regulated way, it's going to be released to the synaptic space and bound to the receptors. These are dopaminergic receptors. And there's multiple types of them, but the message here is that it's a highly regulated process. Why? Because the cells which are receiving this innervation, they're called the medium spiny neurons, are controlled by cortex and thalamus. And they flow down into the globus pallidus and other areas. So it's all connected. So all those wires are for a reason there. So in AADC deficiency, that's not present. In other words, we don't have any dopamine being secreted out or transported. It's just tons of levodopa. It doesn't get converted. So those synapses are empty. Now, if you go and, like in Parkinson's disease, we have no choice because that pathway that brings those fibers from nigra and VTA, they degenerated, they're not there. So even if Parkinson's, we put the vector into this part of the brain, it's not going to do anything because that highway is gone. So that's why we target the receptor cells, those medium spiny neurons. The problem with that is that they don't make dopamine, right? So those are not dopaminergic cells. So you're going to have AADC that's going to convert, just in Parkinson's disease, whatever levodopa, we have to give patients levodopa. They have to get a pill. AADC children make a lot of L-dopa, so that L-dopa is going to get into those postsynaptic cells and get converted to dopamine, which is great because they are some of the internalized receptors. But the minute that dopamine gets secreted out, it's immediately converted by MAOB. So you're not going to have much dopamine floating in the brain. And when you combine it with a very focal delivery, it's a recipe for dyskinesias that may last for a long time. And we've seen it before with the fetal transplants, for example. So we take fetal tissue, put it in Parkinsonian patients' putamen, create little islands, they become grossly dyskinetic. So if you don't have a good spread of the AADC within the putamen, you run into the problems of those hotspots, which can be, I think, very problematic. So we believe that if we use that axonal transport and get AADC made in the cell bodies so they can package dopamine correctly and transport it and secrete it into the synapse, is what needs to happen. I mean, that's going to be a normal situation, at least for the dopaminergic system. So I remember visiting Taiwan and talking to Dr. Lee, and he showed me some images that he generated in Taiwan with, with patients with a couple of tracers, and there's actually normal subjects here. This shows you the normal dopamine production in the normal individual, again, because they have AADC, so they can convert this levodopa, which is being labeled, so you have this being imaged. And then if you use the spec with what's called the dot scan, that actually binds to those postsynaptic, but also presynaptic uptake sites. So this is the most important one, because there you can really see, is this pathway still there or not? It was a critical question for us, and again, I want to acknowledge your help with some of the initial things that we did. So here's some of our patients that you can see on the PET. There's nothing there, right? So again, you've seen it before. I mean, this was expected in the patients. However, using the SPEC imaging, you can see those fibers just normal levels. Again, telling us that that pathway is still there, which is obviously very critical for us to understand that the way that we like to do the gene therapy actually would work well. So here's a couple of points which I want to make, and again, I don't want to extend it too much, but tell you a couple of main points here as it relates to this. So this is a monkey substantia nigra and the VTA. And you can see those little arrows, they basically point out where this stuff goes, right? So it goes in the very specific parts of the brain. And Susan Haber has done beautiful work tracing all of this, and you can see that this part here on the VTA in the red will go to accumbens, and also this part of the VTA and nigra, which is more dorsal, is going to go to the caudate and putamen, and you saw this monkey tracing that I showed you with the gene therapy. So that's basically what it is. So if we target this region, we should be able to have a very diffuse, complete coverage of caudate, putamen, accumbens by normally existing pathway. The other thing which is critically important is the fact that each one of these little cells has that many branches outside. So if you get not even 100% of the cells, you can be assured that they will spread it through multiple cells. So in other words, one dopaminergic cell can terminate on hundreds of thousands of cells in the target area. So it's a highly efficient system. On top of it, if you think about how all of this is being designed, we already talked about dopamine, but there was some mention about serotonin, which is dramatically reduced in Parkinson, in AADC deficient children. But here you have some of the things which are being controlled in addition to motor by dopamine in the brain. And you can see the reward, pleasure, motor function, of course, fine-tuning, compulsion, and also some of the autonomic functions, which we were very interested if this would get affected by gene therapy. What we don't do, it's serotonergic system. We're just not there yet, but definitely we think that serotonin needs to be addressed in those patients as well. So here is some of the setups that we have for doing. So I'm going to show you a couple of cases how we do the operation. So for that we need what's called the hybrid MR-OR, because part of the surgery is done in the OR, and then we can bring patients into the scanner. You can see one here at UCSF. This is the one in Warsaw. So it's all designed in a way that we can really do things very efficiently in terms of reducing the surgical times, but also be able to use both technologies very efficiently. Here's one of the patients. This funny-looking incision is only done so there is no problem with the hair growth. So we have to obviously make a skin incision, but then this grows very nicely and you don't see any issues with the hair. Again, important for women, of course. And here's the coil that is used when you pin the patients into the table that can go in and out from the MR and the OR, which is very critical. We have to be able to see the brain. So this is special coils which we designed for that. Here is some of the aspects of the surgery. We remove tiny little part of the bone, and then we attach these plastic frames that I already showed you. So remember, we're going to VTA and nigra. These are tiny areas within the midbrain. They're really very small. So targeting and accuracy, it's incredibly important. But also, we do want to assure ourselves that we're not just looking at somebody getting gene therapy and not knowing what's happening. So that imaging was very, very critical. So here is one of the patients that is receiving gene therapy. And you can see substantia nigra right here and complete coverage within that structure. And that is shown in multiple different planes. But again, the idea being that we got the volume right, we understood the delivery speed that had to be used to really get good coverage. And then you have now VTA, which is just 2-3 millimeters away from there. See the accuracy here. Also see the fact that there's absolutely no leakage into the CSF space because we can totally control it. We'll just stop infusing if we think that stuff will escape. So infusing AAV2-hAADC vector here with the tracer. Here we have the other side. So they are done sequentially, one after another, with just one cannula, because the infusions are so fast that we really didn't want to do bilateral infusions. And here's the VTA on the other side of the brain. So I think this is really great because we did get in all of the patients just beautiful distributions. Here are some of the data which we are very excited about, and that shows you now the brain. It's a different cut, it's called the Axial Cuts. So here is the midbrain right here. Nigra, it's right here. Before, there's during the surgery, and there's after. So now you can see those two hot spots, which is our expressing gene, and happen in every single patient. So again, we have a complete monitoring of the fact that we did get expression and we got it in the right place. So again, this is where we targeted. So what you're seeing is basically cut right through here. And the question is really, did we get anything in the projection area as I advertised, right? Because we knew that normal monkey, we can get it. We did develop data which suggests that the pathway is still there. But was that true? So here you have an example of the fact that now this projection from here beautifully delivers AADC into putamen and caudate nucleus. So you have a complete restoration now of the innervation tree. In addition, we obviously have it in accumbens, and we believe that some of the activity here in the singular gyrus might also be related to the activity that we see. And again, this is very consistent with all the patients. This slide doesn't really show very well, but the subject number 1 being the least affected in terms of the distribution, all the other subjects, and we have 5 of them now, just didn't put the data here. So in terms of the coverage, we miss on 1, and it's shown here, and that is because of the perivascular leakage. So we are able to see that, right? So we saw basically this leakage from the infusion that allowed us to at least understand that this infusion was not perfect. The others were, and this is the data. We did get almost complete coverage in substantia nigra and probably 80 to 90% coverage in the VTA. Remember, VTA being such a small medial structure, we used almost half of the infusion that we've delivered there. The other thing that, of course, is that the PET scanning is fantastic. I think we've learned that with the putamenal infusions, the signal doesn't tell you anything about the distribution. 0. That doesn't correlate at all. But in our case, I think having this data that really shows the terminal areas, I think it's very telling. The other thing is, of course, what happens to the metabolites, because as you heard already this morning, this is a very good biomarker in terms of success of the gene transfer. So just this one slide, let the rest Toni is going to show in a second. So these are the first 5 patients. You can see the baseline. This is HVA. So that basically tells us that dopamine was formed in the brains of those patients. so very low. Baseline KIT has done all the analysis on those samples. You can see that within 3 months, we have very uniform increase of the HVA levels, which is very reassuring that it all looks the same, except this one patient, which we don't understand. The PET is highly increased. KIT has run the samples twice, so we really don't know why that has not increased. But all the other 4 patients, you can see how dramatically this increased. And it's also consistent. Assuming that this one data point here, it's some kind of fluke, we're going to know this at 3 months. If that persists, the patient has clinically improved. So we have some doubts about that particular sample. And the levels are consistent. They are twice as large as in the Taiwan and Japanese study. In one patient, they keep increasing. So it's early on. I think we're going to follow those patients. The other marker, which is basically tells you how much levodopa is in the brain, a little variable. This one patient that has actually increased. In all the others, it's nicely coming down, which is what would be expected, because if you have enzyme that converts now L-dopa to dopamine, and we see it on the HVA level, I think that should also be visible on the 3-IMD marker. So I'm going to stop right here. I think we're going to have questions in a minute. And Toni, if you would like to show the clinical results. Are you wired?
I think so. Yeah. Can you hear me okay? Yes? Okay. All right, so I have the pleasure of presenting some of our clinical data. What I'd like to do is describe both a summary as well as some individual observations, really for each of the patients that have come through the trial. Sorry, I feel like this is hollow, so I'm going to do the same thing. Yeah. That's better. And so I remember speaking to this audience, I think maybe 2 conferences ago, when we were talking about, what is a clinical trial? What are we aiming to do in a clinical trial? And there's really two big questions that we're trying to answer. First, is the experimental therapy that's being given safe? And the other is it effective? And those are both important questions that we're trying to answer. And certainly for the early stage of this, we had a big responsibility to demonstrate the safety. So that explains a little bit in our protocol. Just wanted to show you what the patients, what the timeline looked like for patients. They came and stayed in San Francisco for 3 months and were closely monitored through that time. First of all, just thinking about the surgery itself, all 5 of the patients tolerated the surgery well. They were in hospital usually for 3 or 4 days, and we didn't have any surgical complications directly, meaning no brain injury that happened as a result of surgery, no reasons to be in the hospital for longer than the expected just post-operative recovery time. And then we were monitoring them closely really through this first 3 months because the gene expression was anticipated to start at some point in the first 3 to 4 weeks. And for those in the audience who are familiar with treating dopamine-deficient patients who might have other conditions, or other children who have Parkinsonism, we know with the use of dopaminergic medicines, there are lots of complicated side effects that patients might develop, from involuntary movements to sleep disturbances and mood disturbances. And so these were all things that we were closely monitoring the patients for in that time. I'll start with the dyskinesia first. I'm going to talk a little bit about specifically the monitoring for side effects, and then also talk about the efficacy or some of the benefits or improvements that we've seen. So dyskinesia, all of our patients did have dyskinesia. As expected, that started usually at around the 1-month mark. And we monitored them by just trying to keep a sort of quantitative score of those movements by looking at the body in all different regions and assigning a numerical score to it. Don't worry too much about the details of that. That graph on the left is more to show the time course of things. So you can see some of the patients were a little bit dyskinetic even at baseline. That's the gray shaded area right on the left. And that was a result of whatever medications they might have been on that could have been already causing some mild dyskinesia. The dotted line in the middle represents an approximate threshold where, scores above that level were things we felt needed to be addressed, meaning typically movements that we would have scored above that line were something that we wanted to intervene to improve the severity of the movements, whether that was tapering their medications, which is usually what happened. In one case, we introduced a new medicine short-term to help to make the child comfortable, and that was some diazepam. But we didn't need any other interventions beyond that. And then you can see the time course there, which is at the peak in severity of the movements for most of the patients was somewhere at around the 2-month mark. And then for the ones who've been followed for longer, we can see that it actually quite quickly comes down. And by the time we get out to the ones who've been followed the longest, now the 2 patients who are 18 months post-surgery, dyskinesia is not really an issue for them. This video is to demonstrate some of the movements that we observed at their worst. So a lot of movements in the face and the upper body. And as I said, the time over which these were, the time period where they were like that was typically a couple of weeks. So we might have a couple of weeks in the middle where we were sort of rapidly bringing medicines down, thinking about whether something else needed to happen. And then by 3 months, I would say all the patients may still have involuntary movements, but they were not ones that were problematic. Sorry, keep going. And then the 2 other things we were really concerned to monitor. One was what would happen to sleep. Now, as you know, some people don't really sleep very well, even at the beginning. And if you just look at the far left of the graph, I would say all but one of the patients in this trial had sleep disturbance as a pretty major problem for them. The most extreme example being the patient who's in blue, who for her whole life basically had 3 to 5 hours of broken sleep a night, was where she started. And all of the others had some degree of that too. A lot of sleep interruption, difficulty settling to sleep, definitely some nights where there was very little sleep. And temporarily that worsened at least for 3 of them. So at the same time where the gene expression kicked in and they started to become dyskinetic, we often also saw patients have a couple of weeks where it was difficult to settle to sleep. And it was also at around that 1 to 2 month time point. And you can see, maybe especially the green and the black, you can see this decrease. This is the number of hours of sleep that they were getting per night on average over a 1-week period. And this little dip that would happen at around the 1 to 2 month mark as well. Then if we follow out after that, definitely sleep has improved in the subject in blue, sleeps much, much better. And several of the other patients also have much more, I would say, continuous sleep, far less waking as well. And together with that, similar time course as well for just mood changes. Now, irritability was a big problem for 2 in particular, for all the patients to some extent, but for 2 of the patients, very severe mood disturbance was one of the biggest difficulties that their families faced in daily life. In one case, on this graph, this is just a very simple graph that shows the number of hours a day that the parents felt that they were uncomfortable in some way. So they were crying or agitated or looked to be upset. And you can see for the subject in red at baseline, that was on average ranging 6 to 8 hours a day where they felt like they couldn't really leave the room. She wouldn't be happy to be by herself. And she just often looked to be uncomfortable, and I'll illustrate that in a moment. And then even if the number of hours a day of being irritable weren't so much, I would say that it had a big impact on many of the families, in the sense that all of life had to sort of be organized around trying to prevent these types of decompensations, so that maybe the child would not leave the house very much, for example, or a couple of them certainly didn't attend school because families felt like that was just too stressful and would trigger a crisis or trigger sort of a mood decompensation. So whether this doesn't tell the whole story, I guess, the timing. I think that the impact on daily routine was another big factor. And this is just a very simple graph on the right to kind of illustrate the parents' perception of that, because we ask them at every time point, how big of a problem do you think this symptom is for you right now? And at baseline, we can see that all five of the families felt that mood difficulties were a problem for them. In black, so the 2 patients in black at the bottom are the 2 families who felt that it was a major problem, and the other 3 felt it was a more moderate problem. And by 3 months after surgery, that had dramatically improved. Actually, the 2 patients for whom it was a major problem, the families then said it was not a problem at all by 3 months after surgery, and for 1 patient, it remains a minor concern. And these videos are to just illustrate that in one of our subjects, because I'm not cherry-picking these videos. I've sort of chose them because I felt they were representative of how the patient's mood really was for a lot of the time. So this is one of our subjects. And really every baseline visit, and I think a lot of the day this was her mood. So it was difficult to tolerate a lot of things. It was difficult for her to tolerate being examined, certainly, and difficult for her family to manage that. Here she is at 12 months. And again at 18 months. Starting to get the development of some head control here. And that's become a stable state for her mood. Sorry. Wait a minute. I'm trapped in this slide. Okay. Another big symptom that we were concerned to observe was people's ocular gyrate crises, because this is a major, major problem for many of these patients. And in 2 of our patients, the ones represented in this graph, had really a large burden of ocular gyrate crises at baseline. These patients would have typically severe episodes that were happening for, on average, a combined time of 8 or 10 hours a week. The other 3 patients had crises as well, but to a less severe extent. And 4 of the 5 patients really had their crises completely disappear at around a month after surgery. And this was varied in timing from, I think, 13 days after surgery in the shortest instance, 33 days after surgery in the longest. So somewhere in that 2 to 4 week period, the crises resolved in 4 patients. And in the other patient who still has them, they did become quite a lot less severe. So we asked families to record, just keep a log of episodes, which I know many families do just as a matter of course, but we asked people to record the time it started, the time it stopped, and how severe it was, meaning is it something that just involved the eyes, or was it something that was a whole body dystonic episode, or something in between. And this, just sort of in more detail, shows the results of that log compared to the summary graph I showed you just now. So here we have for our subject who still continues to have some crises. At baseline would be typical sort of 10 or 12 hours a week of mostly severe episodes. The red arrow at the bottom represents the time point of surgery. And then we saw in the first and second postoperative month the severity and the duration really of the crises come down, when then she's settled into this fairly stable state where now her crises are always mild, meaning they only involve the eyes and not the rest of the body. still continues to have a significant number of them. I mean, she has them several times a week, but milder episodes that are not as prolonged in all. And then I'm going to briefly also mention some of these other motor symptoms before I move to discuss the motor changes, because it's come up in a number of talks today, actually. One thing being just feeding difficulties and it being a prominent problem for a lot of people. It was a big problem for two of our patients at baseline. So 2 families identified feeding problems as being one of their major difficulties. And they actually have resolved after surgery, and I don't have a good explanation for why that would be, but I'll put that out there. And then the other autonomic symptom is the excessive sweatiness, which 4 out of our 5 patients had at baseline, and in 3 cases, it was a major problem that the family said needed 6 or 7 clothing changes a day, something along that order. And that has certainly, in the 3 patients who had at this time, followed out to a year or more, that had disappeared as well. And so our experience so far has been that motor function has visibly improved in all of our patients. The pace of change is very variable, and I'm going to try to illustrate that here and kind of walk you through the timeline of it a little bit. So for example, let me start with one patient who was very hypotonic with poor head control at baseline. And then at 3 months, we can start to see some improvement in spontaneous movements and starting to be able to independently lift his head. Watching Mickey Mouse cartoons. Okay. Whoop. Do you know where'd your screen go? Somehow got out of it. I think I just got out of the presentation screen somehow.
Yeah, don't touch it.
Oh, it's on the other screen.
Yeah.
There we go.
Yeah,
okay, that's all right. All right. Similarly, this is another subject kind of at baseline and the 3-month time point to illustrate, improvements in tone and head control. So at baseline, difficulty with head control. And generalized hypokinesia. And her mom's helping her to sit up because she needs that support. And this is the most recent home video we've received from the family, which was, I think, shortly after her 3-month visit. They were excited to share that she was being able to sit up with quite good trunk control in her seat there. And just as an aside, we find one thing that we've seen really in all subjects after surgery is this tendency to really put their hands in the mouth a lot. That's something that's happened to all of them. At the time they start to move, it's a response that we've seen all of them have. Here's another subject at 12 months of baseline first, very hypokinetic, able to generate very little spontaneous movements on his own. And at 12 months, also starting to develop better head control. You can see him sometimes sit forward in his chair, and with some purposeful movements as well, which you'll hear his dad explain.
Yeah. It's like he's really protecting himself. Yeah. He's scratching his head. He likes to use his hand to his... Well, he already likes doing that, but he realized he can take off his socks by doing this. Okay.
So I hope those kind of illustrate kind of the variability in the time course of how things improve, and that it's a very steady, kind of gradual process that varies in rate from one patient to another. And then out of the 5 patients that we've enrolled so far, one of them has had, a really dramatic and I would almost say unexpected improvement. Here is the final subject at baseline, similarly very, very hypokinetic. Okay. And she developed good head control, I would say, by about 1 to 2 months after surgery, so that came quite quickly. And then after 6 months, somewhere at around 8 or 9 months, she really sort of took off in her motor development. So here she is at 9 months. This is a home video with her wanting to demonstrate how well she could sit independently, starting to use her hands and starting to speak and take some steps. And finally, this is our most recent evaluation of her in the clinic. This was at 18 months.
Wow.
Started to eat as well, so she's demonstrating her new skills for picking up Cheerios.
Very good.
And finally, another home video shared by her parents to demonstrate progress she's made with her walking. So I'm going to turn the mic back to Chris. In just a second. There we go.
Yeah, thank you, Toni. So this is where we are right now, and we definitely have some ideas how we want to move forward. So I think what I didn't talk to you about is that our first surgery took 11 hours. The last one is probably around 5, and we want to go down to 2. So in other words, there's got to be a better way to do this procedure for us, and I think we're definitely developing this. So we also didn't tell you that the first 3 subjects got the low dose, the other 3 receiving the high dose, and all the consecutive patients will receive that high dose, which is just a higher titer of the vector. We also have been testing now a alternative way of delivering the gene as well. So we may also change some of the infusion parameters. So that's, for example, one of those is this scalp-mounted clear point. We don't have to make a burr hole anymore. We can just do everything through the skin. So we've been doing it quite a lot in Parkinson's trial right now. So, so there is no need for the skin incision even. So this is all done really just on the almost automatic way, so it's minimally invasive. We definitely, on different levels of discussing with FDA, they've been really helpful with guiding us through the process. I think this is something that I think it's a continuous and ongoing process. I think the big development for us really has been the partnership with Columbus Children's Foundation. Our plan really is to give this treatment to a foundation. So basically, it will be available to hopefully all the patients around the world on the not compassionate basis, but basically without really registering the product as such as you may think about the gene therapy products. First, we restrict into 5 years. Now we can go below. And then with the new equipment that we have now in place, we can probably treat patients which probably are 2 years or even younger. Also with the MR guiding. So again, this is very important, I think, to treat patients as early as possible. Conversely, we feel very obligated to offer this treatment to older patients over 18 years of age. We are restricted by the clinical trial and the FDA-approved protocol now to enroll patients below 18 years of age, but we definitely would like to start thinking about how to approach older patients. We just opened a second site in Ohio. So we're scheduling first actually international patient, a Canadian patient that we'd like to treat hopefully within 2 months. And then mid-December, we'll treat the first patient in Poland. So we'd like to start treating European patients, starting with patients in Warsaw. The center that I showed you, we started it 5 years ago, performed multiple MR-guided procedures there, Parkinson's patients, and brain tumor, and other things that I also am involved. So I think we will be ready to start treating at least the first 2 patients in Poland, and then we talked with Manju and I think other groups in Europe that we would like to, with the help of the foundation, to basically be able to offer this treatment to kids here in Europe. This ball array stuff is basically another device that we've invented to allow us to do those procedures much faster. So that's the plan. Happy to I guess discuss it as we get to the discussion portion. I know there are changes to the protocol, and I'm sorry, the program. One of the patient parent couldn't come here, so we actually created a video with her giving pretty extensive interview. So Lisa wanted us to show it. This is for the fundraising purposes. So that's not really meant for the meeting here, but I think you will get some of the perspective from the parent on the experience. There's actually two parents of the. This is the first and the second and the fourth, right? Yeah. Can you increase the volume? Okay, can you fix it? The volume here on my computer doesn't work. I don't know why. Okay, so I put the mic here. Where is the microphone? I mean, the speaker is here? It's not on mine, no. I think it's on the monitor, perhaps. Yeah, it's maxed out, so.
Usually the kids after the first month they hold the head up. So then
Can you hear
2
it?
months later, actually she's still not holding her head up. She's really not really moving at all. You know, sleep and drink the bottle, then sleep. You know, it's it's really quiet. You don't really notice you have the baby at home. She's crying, you know, she has a tear. She opened her mouth, but she cannot make a sound. It is so hard even for her to make a sound. In the hospital, doctor said, how often you suctioning her? It's I say, yes, it's not how often, it's never stop. You know, I take the suction machine with me, going anywhere, even go, you know, grocery store. You know, I probably forgot my wallet, but this thing it never forgot. It's always there.
So an ocular gyrate crisis is a very distressing episode to watch. Their eyes are looking up or to the side, and then while this is happening, they can also have stiffening that involves the whole body, and sometimes also changes in their breathing and sweating, and those episodes can, in many cases, last for hours, sometimes 8 or 10 hours at a time.
I was so happy finally, you know, has the diagnosis. I thinking it is, we can get the right medicine to treat this.
They're generally medicines that are often used to treat people with Parkinson's disease, but children with AADC deficiency typically don't really improve much with those medicines.
It's really like a home hospital. We have no time for ourselves at all. This disease has no cure.
At 4 months old is when everything changed with him. Jace had no head control. He wasn't able to play with the toys that a normal seven-month-old kid would play with. He started crying, constant screaming. The lights bothered him, and he would tense up really tight. It was very hard. I had my mom here to help me though, so I was able to, some of the nights that he would be up all night, she would come up and help me. And so then I would sleep for about an hour or 2. So at 5 months, he had been through 3 spinal taps, an MRI, multiple CT scans. We had to do trial and error with a lot of them. At one point, he was on 12 medicines 3 times daily. But finally, once we got set on the correct medicine, he started opening his hands up a little bit more. The crying slowed down a little bit more.
There was no family gatherings, and we'd always try. We'd take Jace, and we'd be the first ones to leave. Be there maybe 5 minutes because Jace couldn't tolerate it. You know, he couldn't tolerate the sounds. He couldn't tolerate the light. He couldn't tolerate a lot. So we would leave. We'd bring him home. The comfort area would be in his room.
Really, he just stayed in his room the whole 5 years of his life. Then what happened is I heard about this gene therapy trial.
It just happened that the program that I've been developing for patients with Parkinson's disease has direct application for children which are born with the deficit of the same enzyme.
In theory, treating something like this is simple in concept because you go, okay, if the gene is missing, we want to just give it back. You know, the technique of giving it back is extraordinarily complicated.
First of all, you know, we did not know if that type of interventions called gene therapy is going to be safe. And we are not ready for children. We just didn't think that we did understand how this system might actually work. So it really has taken us probably at least 12 to 15 years to really realize that, you know, we read it.
Families had to travel to San Francisco from wherever they lived in the country and make several visits. They first had to make a screening visit to come and have some tests and meet the study team, and then once we decided that a child was a candidate for the study, then they would come back to San Francisco for the procedure itself and stay for 3 months.
There was an immediate change in her spontaneous movements. Her head control had improved, her eye contact, her visual focus, her tracking had improved.
Before surgery, Jace could never really open his eyes. I thought he had brown eyes for 5 years. About 2 weeks after surgery, he was able to open his eyes up fully, and he has sky blue eyes. They were huge and blue.
Breathing, that is natural for her now.
Jace is sleeping, we're all sleeping, everybody sleeps all night long. And now he's only in his room for nighttime.
She doesn't need a suction machine anymore. It's a huge difference.
He's having OGCs 3 times a day, sometimes every day, and he hasn't had an OGC since June 2nd.
We feed her by mouth, and she's eating pretty good.
If she is happy, she smiles and says, yeah, in the morning, and like the words she likes to say. And if she's upset, she like grabs my hand and slaps my face and calls me poo.
He goes to school 3 times a week, and he'll go for about 4 hours now. He loves going to school. He's really happy at school.
She's learning how to walk.
So we put the music on. Always can cheer her up, or sometimes she is not in good mood and it can calm her down. It is one day I just saw her. She is moving. She's dancing. You know, it's really amazing moment.
He's more alert of who is in his life and the surroundings of his life.
Her sense of humor, I've always thought it to be very slapstick. After the procedure, she would specifically, very deliberately and voluntarily use her arms for pick up and drop on the floor. It was very funny watching people pick up objects that she's thrown on the floor.
So it's early days for our trial, but we're very encouraged by the early results. We'd definitely like to continue this work and be able to treat more children. Eventually be able to make a diagnosis in a young baby so that they could hopefully never go on to develop all the difficult symptoms that all of our patients experience.
You know, am I building a bridge to nowhere, or is it going to be so expensive no one can, you know, buy a ticket to get on this bridge? You know, maybe there's a place for the non-for-profit biotechnology, or gene therapy company, or a foundation. We'll be very happy.
He smiles all the time.
It's almost like we've gained a whole new Jace.
It is a new life. It's like she like a newborn after surgery.
Mommy?
Mommy. Yeah.
So I think that applause goes to Toni and Lisa Kaplan, who actually helped to make this movie. I mean, so anyway, it's work in progress, and I wanted to ask Javier maybe to tell you about the foundation that, you know, we hope that we'll be able to fundraise for a lot of activity as it relates to this trial. And I think he's well positioned because of the vector production as well. I have a microphone for you here, because I think I'm done.
Thank you, Lisa, for the invitation to be here. And this is a story for us that started 18 months ago, we created a foundation in Spain with the idea of helping kids with therapies that were not accessible for them. We started working on kids with brain tumors, and so far we have taken 15 kids from Spain to other places in Europe to treat them. And we wanted to continue doing things because we were very exposed to gene therapy. We work a lot with gene therapy. We have done a lot of work, investments in gene therapy, and work with a lot of people around the world on that. And we decided that for us, we saw a number of diseases that were totally neglected, and nobody was going to be really paying attention to that. And we have all the means to make that thing happen. So we work with people in the US, Dr. Jules Samulski, Sheila Michalik from Hasbio, and we agree and committed that we are going to help on those diseases by putting all the resources we have, all the expertise we have to solve those. We were exposed at the time to the work that Chris was doing and Toni with AADC, and we decided that that was going to be our first project that we were going to take under the foundation. That started back in April this year, I think, was when we started talking about that. And we are engaged right now in fundraising. We're engaged right now in setting up the awareness of the disease and also getting funds to complete whatever we need to do in order to get the drug registered and get the drug accessible by families. Helping with the China trial, as Lisa asked us, is a pleasure. So anything we can do will be done. We have 3 fundraising events coming next week, and the following week in the US. We have a fundraising event in Raleigh, North Carolina, in Washington, D.C., and in Naples, Florida. And one of the things we do, we use music for those things. So we use music as the universal language. So we have a number of well-known artists who are committing their time and effort, and we put concerts and we use them for fundraising and raise awareness. So our commitment is there. We have also the benefit of accessing gene therapy manufacturing through the partnership we have. So for this may not be necessary because Chris has enough vector for this one, but for other diseases we'll be able to provide vector for the development. We have other ideas of other diseases we are going to treat. So for us it's a real pleasure and something that it really come like a dream to be able to help. And the work, and we have seen some of the families, and being with you this week is going to be phenomenal, and really you have the commitment that we are going to help as much as possible. Well, thank you.
Thank you very much. Now, are you okay? Now, Toni Pearson is going to continue with the presentation of the natural history of AADC.
Thanks. Yeah.
Thank you.
Hopefully.
Yeah, it's coming.
Do you want me to move your microphone a little bit further up? Is that
So next step is to change the doses.
I can probably just try to, I think we probably maximize I think AADC saturates. So I think we want to still improve on the distribution in the midbrain.
Okay, well, I'm going to try and get us more or less back on time because this should be a short and sweet talk, but I wanted to give some interim, I guess, an update on this topic. I spoke at this meeting a couple of years ago on this topic of trying to improve our understanding of natural history of the disorder, including, information about symptoms in patients at an older age. And I just wanted to come back to you with some findings that we've had in the meantime, because we have actually had a number of families participate in a questionnaire on this. Very briefly, a natural history study describes any study that attempts to assess how a disease evolves in an individual and in a group of patients over time. The type I'll be talking about today is what we call retrospective, meaning that if we start at the time point of now, it's collecting information about things that happened in the past up until now. There's another type another main type called a prospective study, where the goal is to, from now, sort of start collecting information from the current time point into the future, with the idea of that being standardized information that's collected and planned in advance. And some of you may be familiar with the INTD registry, which is an effort that several of us in this room have been involved in, and that's the type of study that INTD registry does, where we recruit patients into the registry and periodically try to update the information and collect it as time goes on. So what I'll be talking about today, there was the retrospective study I did, which was basically a survey or questionnaire. And the responses were through two sources. This is a long-time collaboration with INTD to make that available through the registry, so that patients who are enrolled in the INTD registry, this was one component of the information that was collected. And that started in 2016, was when we got that up and running on the INTD site. And then more recently, just this past year, I know a number of families would have received 1 or maybe 5 or 6 emails from Lisa requesting that they complete an online version of that exact same thing that we were doing through the INTD registry to try and collect that similar information. So for patients who were enrolled through the INTD, let's start with the AADC Trust, actually, because that's my first bullet point. So this was something that was completed by parents if they were recruited through the AADC Trust. For patients who were enrolled through INTD, then the information would have been entered by the study team or the doctor of the person that the patient saw at their hospital or clinic, probably in collaboration with the families, I would imagine. And it was the topics that were addressed in this certainly concerned things that we felt were already fairly well characterized in the literature, things like how to diagnose it, what are the initial symptoms, age of onset, a lot of things about how the disease presents. And some of the additional things we wanted to gather information about were how do people respond to treatment, what are the side effects of treatment that people experience, how did the different symptoms evolve over time, and what other medical issues come up for these patients in addition to maybe the ones we consider the primary disease symptoms. Also looking at just developmental outcome and activities of daily living. And there were also some questions there about end of life, for a number of families actually responded to this questionnaire who have lost their children as well. Altogether, so far, we have received responses from 64 families of 64 patients, and that's combined through the sites. So 48 parents directly responding after being contacted through the AADC Trust, and 24 enrolled through INTD with a little bit of overlap. So some patients actually were recruited through both mechanisms. And 39 of them were female, 25 were male. And I have to thank everybody who contributed to that from the family perspective, because I think around about 2/3 of the families that were contacted through the AADC Trust actually did respond, which I think for a survey of this type, I think is a very, very good response rate, and is really a kind of testament to the community here. It was quite a diverse sample. We had participants from around the world. Altogether, it was 23 separate countries when we count everybody. Roughly just under half European, and then Asia, North America, and South America as well. And a little bit in contrast to when we look at just the existing literature of patients who've been reported, a lot of them the tendency has been for them to be young. So more than half of the patients that are in the existing literature are under age 5, at the time that they were described. And this cohort is a little bit older. We sort of see a shift. There's still a bit of a peak in the early ages there. This shows the age of the patients that were in this study, and the number at each age. So we can see there is sort of a peak in that early childhood period, but there's, as you can appreciate, there's patients of really across the age spectrum, and dividing people into categories of age. We saw sort of had the age 2 to 5 was the biggest group, but also, a fairly good sampling from across the age spectrum, including adults. And so one of the first initial things I looked at was just reported age of onset and time to diagnosis. So the median age of onset, so it was 3 months, so this is an early in infancy presenting condition generally. Almost all the patients actually were under 6 months of age at the time that their parents thought their symptoms begun. Only 2 patients in this entire sample reported their initial symptoms to be after age 6 months, and it was at around 1 year. And then the graph on the right just shows latency to diagnosis. So latency to diagnosis meaning the age where the symptoms started to the age where the diagnosis was made. And what we can appreciate is that on the left half there, the patients in this study who are under 10 years of age tends to be a diagnosis that's made much more quickly than it used to be. So the latency to diagnosis being under two years, and in many cases much less than that for patients in the last decade. In contrast to, if you look at patients over age 10, who often had years to time of diagnosis. This graph just lists frequency of initial symptoms that were reported. Now, it doesn't mean that if somebody doesn't necessarily include all symptoms that the patients had, in the sense this is what either the doctor or the parent recognizes as this thinking of what their initial clue to the diagnosis was. Hypotonia was the leading one. Approximately 60% or just over reported that as their first clue. I would suspect that the actual prevalence of hypotonia was higher, but it was the most commonly reported initial symptom. And then ocular gyrate crises, delayed development, feeding problems, all common kind of initial symptoms that were identified. And ocular gyrate crises really were experienced by a majority of patients across all ages. This just shows the percent of patients that reported having crises across different ages, and we can see there's actually 100% of patients aged 2 to 5 in this cohort that said they had ocular gyrate crises, so it's very much a feature of early childhood, school-age years. And they were less prevalent as people got older, but even if you look at the adult respondents to this survey, it's over half of adults, or over half of the over 18s that still reported having ocular gyrate crises. And the frequency of episodes was 3 or more episodes a week in nearly half of the patients, and tended to be long in the majority of patients as well. In over 60% of patients, episodes were typically over 4 hours in duration. Another symptom that was interesting to look at here was sleep disturbances, which were one of the common early symptoms. And here the graph shows the prevalence of excessive sleepiness is shown in blue and insomnia in orange. So appreciating that in children under 2, it was much more common for people to report excessive sleepiness being a problem as opposed to difficulty falling asleep, although both things were there. And then you can see that really changes at older ages, where older children were much more likely to be reported as having insomnia as a major problem than being too sleepy, although once again, both things could be present. And then developmental outcome. This is, I think, maybe one of the things that's the hardest to to gather from really any paper that you read about AADC deficiency, because as I said, many of the papers describe younger patients, and it's also hard work to kind of assemble a lot of case reports and get an impression of the whole group. So in this study, we were able to look at that, and looking at just some main motor milestones, we had about 30% of parents or doctors reporting that the child had some head control, although it was typically delayed in its development. So 3 months was the youngest age reported to have it attained, but that extended all the way up to 4 years. 18% of families reported that their child could sit on their own, which is would be higher than I expected. And then also looking at the slightly older group of children, excluding the very youngest ones, also 19% of this cohort reporting independent walking and 15% able to speak. Another question that we asked was actually about loss of skills, because this is something that I think we don't typically focus on very much, and I think anecdotally perhaps all of us have heard from families about about skills that were felt to be there at one time and maybe fluctuated or were lost at a certain time point. And 24% of the families here reported that there had been some loss of skill, and it was typically something that would happen quite early. In some cases, it was able to feed a little bit at the beginning, but then stopped being able to feed. In others, it had gained some head control and then lost that ability or lost other independent motor skills. And also in a couple of patients reported that they had some speech, were able to say some words, but at some point that regressed. And then I think this might be my final initial thing to share. I was just also looking at sort of daily life, activities of daily living, and trying to get some sense of independence for moving around, both in the community and at home, routine daily things like eating, drinking, bathing, dressing. So of the respondents in this study, we found 11% of them reporting that they were fully independent for those things, 19% some independence, and then 70% with severe impairment requiring, full assistance really for all of those activities, which would be in line with, Manju spoke this morning about the consensus treatment guidelines that were published last year and did do a very extensive review of all previously reported cases. And I think these findings would be in line with what was implied in that document. And then also just looking at school and work. So school attendance was almost 2/3 of people who would be school-aged. And of the ones who wouldn't, I think it would be interesting to explore a little more why they were not in school. I think it's usually sort of medical fragility or symptom burden or worry about getting sick. But that's information I need to get more information about. And then finally, of the 10 kind of older people in this cohort 2 out of the 10 of them, so 20% in that small sample size, actually were working as adults. So that's a very, very quick preliminary look. We're still, I haven't quite analyzed everything yet because I think we will still expect, there's a few more patients who need to be added in. And some specific things that I want to look more closely at are sort of medication benefits and side effects, associated medical problems, and a little bit more detail as well about survival, mortality, and end of life. I want to thank Kathleen Black, who's my study coordinator, and would not have gotten the online survey up without her. Lisa Flint, of course, because it wouldn't have been possible without the AADC Trust. And it also wouldn't have been possible without INTD. Thomas O'Bladen and I have been talking about this for a number of years. And he and Katrin Jelch and a number of other doctors really helped with translations, with recruiting patients, and the patients in this study came from 9 different INTD sites. So I thank them as well. And with that, I think I'm done.